Hello everyone, and welcome to phyloseminar.org, a project supported by the Society of Systematic Biologists. I want to remind everyone before we get started that there is this Q&A app that should be on the side of your screen, and you should put questions in there for Matt. So today is the first talk in a series of 3 talks on ancestral recombination graphs. Today we're going to hear from Matt Rasmussen, now at Counsyl. Then we're going to hear from Peter Ralph from USC, and finally from Matthias Steinrücken speaking from his new position at UMass Amherst. Matt did his undergraduate at the University of Minnesota NCS, then his master's and PhD at MIT under the supervision of Manolis Kellis. So if you haven't checked out Matt's work from this time during his PhD, I definitely encourage you to do so because it's really nice and I feel a little bit underappreciated in the phylogenetics community. It's on species tree reconstruction in the presence of duplication, transfer, and loss, which is, and lineage sorting. So, sorry, in presence of gene duplication, loss, and lineage sorting. Do you have that right now?
Yep.
Okay. So that's a really hard problem, of course. So then he worked with Adam Siepel at Cornell, and then started working on the related problem of inference of ancestral recombination graphs, which is what he's going to be talking about today. All right, now. Thanks. Take it away.
Great. Thanks again for giving me an opportunity to talk. I've been a long-time viewer, first-time presenter of the Phyloseminar series, yeah, I'm very excited to be participating in this particular series. So the title of the talk is Mathematical and Visualization Tools for Working with Recombination Graphs. And Eric had asked me to spend a little bit of time on introducing what ancestral recombination graphs are. They're a data structure, and they are very powerful for representing ancestry within population genetics. And a lot of the different features that people think about, like local trees, genealogies, identity by descent, time of the most recent common ancestor, can be thought of as summaries of the ARG. So understanding the ARG very well and can give us power for inferring all of these different summary statistics that people are interested in. I'll also review the most common stochastic process that people think about when they think about ARGs and think about distributions of ARGs. It's called the coalescent with recombination. And then more recent work has been on approximating the ARG. It's a pretty difficult data structure and stochastic process to do with directly. A lot of calculations are quite expensive. And so more recent approximations that view the ARG in a sequential way and also with the Markovian assumption, that's what SMC stands for, Sequential Markovian Coalescent. They have been quite powerful, and they underlie a lot of recent work in terms of inference and simulating very large, realistic sequences. The second part of the talk, I'll talk about some of my work that I did in Adam Siepel's lab that was published earlier this year on inferring ARGs. So given sequences from several modern-day individuals that are undergoing recombination, can you infer the ARG that describes their ancestry? And so that was a piece of software called ARG Weaver. And lastly, I wanted to include a few slides on visualizing ARGs. I had found in my own work being able to better visualize ARGs and think about what structures are occurring, I found that to be quite useful. I think there's quite a bit of work still needed to be done there, and so I thought I would just pose a couple of interesting visualization problems. It could be used to look at a lot of things. So first I thought I'd quickly introduce how I got interested in ancestral recombination graphs. As Eric mentioned, my PhD work was in phylogenetics. I'd worked on a lot of algorithms on working with binary trees, and I really liked that that rich combinatorial data structure. I really liked the working with the probabilistic frameworks around thinking about the distribution of phylogenetic trees and things like that. And for my postdoctoral work, I was thinking about, what would be a new topic, but still have those kinds of properties that I like. And I came across this review paper by Adam Seppel in Genome Research in 2009, and he was talking about population genetics, but he was describing how even in population genetics there is a equivalent of the phylogenetic tree, just kind of like a souped up version of it that can also represent recombination. And that's called the ancestral recombination graph, and just showing a lot of the figures from the paper, you can really see that there's a lot of rich information there, and so that was kind of maybe the very start of my interest in the problem. And it turned out that for my postdoc I ended up working in Adam Seppel's lab. Okay, so why are they so cool? Why are they important? In many ways, you can think of the ARG as the sufficient statistic for a lot of things that you would want to infer. If you knew the true ARG, and that's a hard thing to do, but if you got lucky and you knew it, you would have extracted all the information that there is from the DNA sequences in your sample. And there are many applications. So if you could infer ARGs, you could infer different patterns of selection, different patterns of demography, perhaps even association mapping could be done more accurately if we could model and infer ARGs better. So just to introduce the topic, I thought I would introduce it the same way that I kind of entered the field, which was from phylogenetics, and I think that's maybe the background of a lot of the audience here. And phylogenetics are the most common data structure is the tree. That's kind of the workhorse for modeling ancestry. And what's nice about a tree is if you don't have to worry about recombination, then it is sufficient. Without recombination, every sequence has one parent, and so that's what a tree data structure is. And you can use this for species that really don't have recombination, or for any small genomic region that is unlikely to contain recombination. So some small non-recombining block, or perhaps maybe you're looking at very long periods of evolutionary time, and the kind of distortions that recombination introduce are negligible. Okay. A lot of work that looks at gene trees along the genome and looks at their differences and models that with incomplete lineage sorting in order to infer species trees. They are making use of recombination. That is what's causing those trees to be different. But they're adding the assumption that each gene is one of these small genomic blocks of roughly no recombination. Okay, so how does recombination change all of this? In a really idealized sense, you can think of recombination as taking two sequences, say A and B. A crossover occurs at some position R along their length. These sequences are homologous or orthologous. And as a result, we get a new sequence C, which we can think of as, say, the prefix of A concatenated with a suffix of B. All right? And then graphically, that would be like a node C that has ultimately two parents, A and B. They're labeled as one being the prefix and one being the suffix. And then also, just to be complete in describing what occurred, we'd want to record exactly the position R at which this crossover happened. Okay, so if we go back to our tree and we try and add in such a node, we would find that a node now has two parents. And this is an orthologous sequence. It's just from another individual somewhere in the population. So if we continue to trace its ancestry back into the past, we'll eventually find that it coalesces with some other lineage in our tree. So we'll connect back up. And if we look at larger and larger blocks of the genome, that block will capture more and more recombinations. So that can just be thought of as additional nodes, each of them adding one more branch that then needs to coalesce into the tree, and they can coalesce even above the tree. And so now we no longer have a tree, we have a DAG, a directed acyclic graph. It now has two kinds of nodes. We have the original kind of coalescence nodes. These are nodes that represent two lineages that go back into the past until they find a common ancestor. You have one parent, two children. We now have these new recombination nodes. And this is a data structure that has been studied for quite some time now. It's called the ancestral recombination graph. And the generative process for how such ancestry is created has been modeled most commonly with this process called the coalescent with recombination. But there's many other equivalent ways of thinking about how this data is generated, and also approximate ways. So I thought I would just briefly review this stochastic process because it underlies a lot of inference and simulation algorithms. The main parameters of this process is that you have an effective population size, how many haploid individuals are in your population. You have, or how many individuals you have in your population, the length of the sequence that you are modeling. That's important because the longer the sequence, the more recombinations that you would have to consider. And then a recombination rate, which you could express in recombinations per site per generation. This is a process that goes backwards in time, just like the coalescent process. And so you start with, say, K lineages at the modern day, say time 0. And then it's a Poisson process of several events. There's two main events we're considering: coalescent events, which takes two lineages and combines them into one; recombination events, which split a lineage into two. That's what it looks like when you look at it backwards in time. And they each have their own rates. So this is the same rate that you see in the coalescent model. And recombination rates are just this rate rho times how many lineages are currently present in the length of the sequence. And this process continues until you get down to one lineage in which it terminates. So just to show kind of an illustration of that, let's say we started off with 4 sequences from the modern day, okay? And if we go backwards in time, we would find that eventually 2 of these lineages coalesce, so that's a coalescent event. We go back a little bit further, and we might encounter a recombination event. So, as I said, this will split the lineage into 2 lineages, and one of them contributed a prefix of its sequence, which I'm illustrating as by drawing the sequence again and kind of graying out the part of the sequence that is not contributed, and the other lineage is contributing the suffix. And then if you concatenate these 2, you get the descendant. All right, so if we continue, in the field, these kind of regions of the sequence that is not contributed, that is called non-ancestral sequence. And that kind of plays an important role in several cases. If we just continue to go back, lineages will continue to coalesce. Maybe some more recombinations occur, possibly at other parts of the sequence. And eventually, everything coalesces. Okay, so there's software that implements this process that you can simulate. The MS software is very popular. It's used to make fairly realistic sequences. And there's work for even inferring these ARGs based on this stochastic process called Lamarck. And it's a very challenging problem because ARGs are such a huge state space. It's really hard to explore. And that was part of my work, was thinking about how to explore the state space even better, even faster, so that we could scale up to very large sequences and large number of sequences. But I'll get to that a little bit later. So I wanted to just spend a little bit of time of looking at the ARG data structure a little bit more, because there's lots of different ways of looking at it, and different perspectives are useful in different applications. So one very common way of looking at ARGs is in terms of local trees or marginal trees. And the idea is, if we consider only one site in our sequence alignment, that one site is still has an ancestry that can be modeled with a tree. Okay, and so just to illustrate that, let's say we wanted to know what the ancestry was of a site chosen in this blue region of each of our sequences. We could trace up the ARG to see where that where this lineage gets its ancestry. And the main thing to look at is the recombination nodes. When we get up here, we just simply ask which of the two parents contributed sequence for this site. In this case, it was the right lineage. For this recombination node, it was the left. And if we just keep tracing up, we eventually see the tree that describes the ancestry at that site. And we could do this again for any site along these sequences. So if we ask for the green region here, we see it's a fairly similar tree. The only difference is that for this recombination node, now we trace ancestry up to the right. And just let's look at the yellow region. We again have yet another tree. And so you can think of the ARG as just the union of all of these local trees. Just another kind of way of connecting these two perspectives. Here I'm drawing again the sequence. I'm again drawing like what parts of the sequence is ancestral and non-ancestral, and that corresponds to local trees. So any part of a sequence that is non-ancestral means that there's no local tree that passes through at that site. So sometimes it's useful to think of it that way. I also just want to cover a few corner cases. So if you implemented a simulator exactly the way that I first introduced it, it would be fairly inefficient, because if you were to pick recombination crossovers uniformly across the entire sequence, you could generate recombination nodes where one of the parents is actually completely non-ancestral. And what that means is whatever mutations happened along this branch have gone extinct. They would not contribute to any of the sequences in our data sample. So a lot of actual implementations of the coalescent recombination do a slight modification of avoiding ever thinking about such branches, because we have no information about them. Another kind of interesting corner case is this. I've adjusted the exact crossovers for some of these nodes in order to make a particular example where two lineages coalesce, and in doing so, you now have non-contiguous ancestral sequence. So what's interesting about that is it can create very long-range correlation in local trees. I'll explain a little bit more about that in a moment. Okay, so let's go back to the original example. We now have the two perspectives, the ARG and all of its local trees. A very common way that people think about the local trees is to pull them apart and view them sequentially, left to right. So now I have the same trees. I've now drawn my four sequences as a sequence alignment. And you can think of the recombinations as defining breakpoints in these alignments. So within each alignment here, say from bases 1 to 204, this is a non-recombining block. Every site in this block is described by this tree. All right? And so on for each of these other blocks. So we can see the trees differ, but they share a lot of similarities, and that's because they are all subgraphs of a common ARG. And actually, neighboring trees are extremely similar, and they actually only differ by single SPR moves, so subtree pruning and regrafting operations. So this is an operation probably fairly familiar to people within phylogenetics. It's an operation used to search tree space for doing maximum likelihood and parsimony and many other methods. But here, the way it's related is the reason why one tree changes is from the other is because of recombination that removes, say, the prefix parental lineage and replaces it with the suffix parental lineage. So we can think of the recombination as tearing off this subtree and regrafting it to the right. And then when we look over here on the green tree, we can see that that operation has been applied. And we can just step along all the local trees, one SPR operation at a time. So we need to be a little bit careful here. When we see these local trees, we see them changing by one SPR operation at a time. It seems to suggest that maybe these trees are markovian. That is that the next tree is only dependent on the previous tree and is conditionally independent of any trees that come before it. But that's not strictly true. There's actually very long-range correlation, and that's due to that non-contiguous ancestry corner case that I described. But you can still write down a generative process in this sequential view. It's not Markovian, but you can still write it down, and that's described in this work by Wolf and Hein. And sometimes that perspective is very useful. Okay, so it turns out that although it's not Markovian, it's pretty close. So how bad would it be if you added the assumption that these local trees are Markovian? That is, the probability of tree I conditioned on all the previous trees is really just equal to only the last tree. And there's been two formulations of this approximation called the sequential Markovian coalescent, and then a slight modification to it based on exactly the way recombinations work. And what's nice about this is that this process is very convenient for creating fast simulators. You have kind of a fixed memory size because you only have to consider the last tree. So then the question is, like, could we also use these models for inference? Could we infer an ARG? Could we infer local trees given just the modern sequences? If so, then we could do a lot of the applications that I previously discussed. All right, so it's a Markov process. Maybe an HMM could be used to do inference. However, unless you do something clever, a naive implementation is going to run into a lot of issues because the state space is extremely large. The number of tree topologies grows super exponential with the number of sequences. You probably also want to consider lots of variations on the branch length, and so that's a pretty large state space. But the idea is a pretty good one. You could think of the local trees as your hidden states in this HMM, and the emissions would be your sequence alignment. So there's been a lot of work on this, and it's been quite successful in a lot of different applications. And you can kind of organize a lot of that work or think about it in terms of what they did to avoid running into the large state space. So you could either use a fewer number of sequences in order to keep the state space smaller. You could think about ways of avoiding modeling the entire local tree, approximations such as maybe trunk genealogies, or thinking about just the first coalescence in any tree, local tree. You could think about just treating it as more of a combinatorial problem and avoid modeling branch lengths. And in my work with Adam Siepel in his lab, we had to make our own trade-off. So our approach was to think about a sub-problem. Could we avoid modeling the entire tree? Could we think about only modeling the ancestry of one sequence at a time and condition on the ancestry of the other n-1 sequences. Okay? So if you could solve that subproblem, maybe you could build upon that to then sample the entire ARG. Okay, so this was the key idea behind a paper we published earlier this year in PLOS Genetics, and the software implementing it is called ARG Weaver. So the key subproblem is pretend somebody has already solved all the local trees for n-1 sequences. Okay, so here I have several local trees. Here I'm drawing one non-recombining block and drawing the same local tree multiple times here. And then we can see that these breakpoints introduce recombination that slowly modifies the tree, one at a time, one SPR move at a time. Okay. So now somebody gives me a new sequence. All right. So I'm going to draw that sequence number 4 is this blue sequence down here. And adding that sequence to all of these local trees is really just adding one more leaf and one more branch to each local tree. And then we have to decide how does that new branch coalesce into each local tree. As we look sequentially, that branch will jump around. It'll jump around because recombinations on that branch break it and cause it to recoalesce somewhere new. Another recombination breaks it, makes it go lower. It can also move around because the given local trees move out from underneath it. So here's an example of like a recombination breaks the branch that this new blue branch is attaching to. And so when we go one tree over, we see now the blue branch has to go much higher before it coalesces. So that subproblem of adding just one more branch to each local tree, we call that threading a sequence into the ARG. So how could we sample that from the posterior distribution, assuming an SMC-like generative process? So first let's look at what happens within a non-recombining block. So here we have 2 sites. The given local tree in black is not changing. We have our new branch, and we can represent basically a state for that branch. And that state is state Y, and it really just depends on what branch X you attach to and at what time do you attach at. If we use, say, discretized time, several different time points, then we have a finite number of attachment points for this new branch. And it's a small number of attachment points. So that's desirable for keeping a small state space in order to have efficient computation. And then this branch, we can think of transitioning from state to state just based on the probability of each SPR move. And so this actually looks a lot like an HMM. Our hidden states are just the attachment point, this vector of Y's. The transitions can be derived from a discretized version of the SMC model. And again, the emissions are just columns in the sequence alignment. Okay, so what good is this HMM? Well, with it, we could apply the forward algorithm and stochastic traceback. And what that would allow us to do is sample a particular threading of this new sequence. That is, we could sample a vector of trees, each of them with n leaves, given a vector of trees with n-1 leaves, recombinations, and the data. And so not only can you sample where the branch occurs, but you can also sample where the individual recombination nodes occur. One kind of interesting thing about this algorithm is that we also have to consider what happens when the local tree changes out from underneath us. And so for each local tree, our state space is different. There's different branches to attach to. There might be even different number of states depending on how big the tree is. And so this is a heterogeneous HMM. This is just kind of a visualization of what like the forward algorithm would look like. For each non-recombining block of sites, we have lots of states which represent the attachment point of our new sequence. There's a single transition matrix for each non-recombining block. There's a one-time use transition matrix that helps you jump from one block to the other. The state space can grow and shrink, but otherwise it's the usual forward algorithm. All right, so that was a sub-problem. We can now go from an ARG of n-1 sequences to n. How could we use that to build up an entire ARG? So you might think about applying that algorithm serially, one at a time. You could start off with a trivial ARG of a single sequence, which is just one branch, and then add one more sequence at a time, slowly building up a bigger ARG. That would work. You'd get an ARG, but it would not be a true sample from the posterior. It's biased. The initial sequences were not conditioned on the full data. But we could use this as a starting point to improve upon. And so the additional idea we added was to do Gibbs sampling. Now that we have an ARG of n sequences, what if we choose one of the leaves to remove, brings us back down to an ARG of n-1 leaves, and then apply the threading algorithm again to add a resample where it could attach. And if we just rotate through all of the leaves, this turns out to be a Gibbs sampler for the ARG. And so if we run that over several iterations, it'll eventually, hopefully, converge to the posterior distribution of the ARG. We implemented that, and it does work, but it can only work up to a certain point. We found that once you have about 6 or 8 sequences, the mixture and the convergence is quite poor. And in retrospect, that's actually seemed kind of obvious. The idea of removing a leaf and reattaching it to the ARG means that it's really hard to adjust the internal parts of the ARG, the very deep branches. You have to slowly work those branches out to the edge, and then you can remove them and resample them. So we thought a little bit harder and thought about whether we could resample internal branches. And it turns out there's a lot of similarity. We could reuse this basic threading trick. Let's look at a bunch of local trees that are the same. So we're looking within a non-recombining block. And let's say we wanted to remove this internal branch and resample its coalescence. If we did that, it would break our tree into 2 pieces. Let's call one of the pieces the subtree, and then the main tree contains the original root and a basal lineage, right? When thinking about how this lineage would recoalesce, the coalescence process is memoryless. It's independent of what happened beneath it. So it really doesn't matter what's going on, what the structure is at this part of these trees. And so if we hide that from ourselves, you can see that this is really just another instance of the threading problem again. It really is just adding one more node and thinking about how it coalesces and jumps around from tree to tree. There is one remaining challenge, and that is how do you do this kind of removal of internal branches and resampling across breakpoints? What happens if the given local tree is changing? And I probably don't have time to go into it too deeply, but we did have a strategy for that as well. And the idea is we wanted to remove a series of branches that shared a lot of common ancestry. It was kind of a smooth transition from tree to tree. And we introduced a new kind of dual data structure called the branch graph, which allows you to look at series of branches that share ancestry. So just to quickly define, here I have a series of local trees changing based on SPRs. In the branch graph, for each local tree, I create a vertex for each edge or each branch in the original tree. So I've numbered the branches here, so I've numbered the vertices here to show the correspondence. And then between two local trees, I draw an edge between two vertices if it's the same branch or if it shares ancestry. So, for example, the leaves, their branch kind of exists across all the trees, so they jump across. Recombinations break a branch, like number 6 here is basically broken because a recombination happens underneath it, and then creates new branches. So that's what these crossing branches do. However, paths in this graph give you branches that are either equivalent or very similar to one another. So it gives you like a very smooth chunk of the ARG to remove and resample. So implementing all of those ideas, we now have actually an MCMC sampler. It's like Gibbs, but there's actually the accept-reject ratio is not always 1, so it becomes an MCMC sampler. And we find that if you run it for enough iterations, various statistics like the joint probability or number of recombinations eventually converge to the true answer. So this is on a simulation. We did a lot of validation of this inference algorithm for lots of different summary statistics. Can you recover the true number of recombinations? Can you recover the overall branch length of the ARG? And we tried this for a whole bunch of different parameter settings. One kind of the most interesting parameter setting to look at was the ratio of mutation rate with recombination rate. So the higher that ratio is, the easier the problem is. The more mutations you have, the more information you have about what the ARG looks like. The closer that gets to 1 or below 1, the tougher the problem becomes. And at some point, you really have very little information to recover the ARG. And so that's going to depend on which species and which regions of the genome this is inference of the ARG is achievable. And so this is just an example of the same plots, but just we looked at lots of different ratios and looked at that you could recover the true value in many cases. Here's an example of another statistic. So if you plot the time to most recent common ancestor across the genome, again, this is a simulation. In black is the true answer. In red is our 95% posterior interval confidence interval for each side along the genome. So you can see that it recovers that. So then we started, after we kind of did that evaluation, we then also looked at real data. And so this was a lot of work by Melissa Hubitz, a researcher and now a student for Adam Siepel. And this was also presented in a paper. So one thing we would like to do is be able to infer regions that are under negative and positive selection. And there are many summary statistics that you could use to detect such regions. For example, we could look at pi, which is a measure of diversity at any site. And what you would find, and this has been described many times, is that coding regions have a reduction in diversity. That's because negative selection has been removing mutations from the population. But you also see a reduction of any sites that are nearby, and that's due to linkage and the effect of background selection. In positive selection, if you look at sites that are undergoing sweeps, say for example partial sweeps, that also reduces diversity, and you also have linkage, but in this case it's hitchhiking that causes neighboring sites to also be reduced. So both positive and negative selection cause these dips. Could we get something a little bit more out of out of the ARG. We now have the local tree at every site. What could we do with that? In negative selection, we would expect kind of the local tree to be overall shorter, like it just scaled down. There's just persistent removal of variation over long periods of time. With positive selection, we would expect the tree to mostly not be shrunk, except for the subtree that descends beneath the mutation that is under positive selection. Its lineages would spread out very quickly, or you could think of it as its subtree as being squished, being much shorter. Could we develop a statistic that picks that general pattern up? Well, if we try to use just TMRCA, we don't quite see, we see a difference. We see that the TMRCA is reduced for coding regions. The reduction does happen at sites that are in linkage with them. And we, interestingly, we do not see that reduction for positive selection. And just based on this cartoon, you can kind of see that, you know, TMRCA is just telling you the age of the root node, and in many cases that root node it outgroups the mutation that is being selected. And so how could we pick up on this distortion? Well, what if we asked about, say, the ratio, the kind of like the ratio of the TMRCA of just the first 50% of the lineages divided by the overall TMRCA. That would try and pick up on the fact that the overall tree is tall, but there's some subtree that is very squished. Doing that here in negative selection, we see that this ratio is not distorted, is consistent across all of these regions, and yet for these partial positive selected sweeps, we see this dip. So that's kind of a nice application of how you could distinguish between these different types of selection if you go through the route of inferring the ARG. Just one more thing that's kind of interesting is with an ARG, you can now place mutations on internal branches of the tree. You can try and estimate when a mutation occurred, and, you know, the age of an allele. And yeah, the age of an allele should be distorted by selection. The more selection that is occurring, the younger we would expect that age. So if we look at mutations across different mutation classes, we should see a trend. And so if you look at neutral regions, the age is quite high. As you look at regions such as benign mutations, they are also quite high. If you look at mutations that are thought to be under negative selection, such as mutations that are damaging, according to Polyphen or according to ClinVar, the database of clinical variants, you see that there is a reduction of age relative to benign variants and neutral variants. And this trend is true within any allele frequency. So, of course, you will see a difference in allele frequency between benign and deleterious variants. But even when variants have the same frequency, the age that we estimate for them show the same general trend. All right. I just want to briefly talk about how to visualize ARGs. So in phylogenetics, the phylogenetic tree is our main way of visualizing the ancestry. We can use the topology to understand how genes or species are related to each other. We can use branch lengths to tell us the amount of time that it has passed. The ARG, you would think we could do the same thing. But if you ever try to draw an ARG for anything larger than a toy example, it's a total mess. The lines cross. It's really hard to get any general idea of what's going on. And what makes it challenging is that there's three important dimensions. We have the generations over time, we have the sites along the genome, we have lots of different individuals. And so how do you represent all three dimensions in a 2D plot. Also, what makes it hard is, as I said, the layout. Trees are easy to lay out, but ARGs, you always have some crossing branches. So in most visualizations, you see one of the dimensions is sacrifice. So this traditional ARG, kind of sites are not really being visualized that well. Here, we're kind of sacrificing individuals. We're kind of reordering individuals from tree to tree. And if you really wanted to track what was happening for a particular individual, it would be very hard to look up how that individual is moving around in anything beyond toy examples. So I briefly thought about how to tackle this, and I thought about what could we do if we basically take this local tree visualization, but try to compress the tree drawing, have it take up less real estate, visual real estate. That is, could you visualize a tree in just one dimension? And so one thing that I found that worked was to draw one dot for every leaf of a tree, draw them vertically, and then let the spacing between each dot represent one internal node. And so you can think of it as the most recent, the spacing between any two neighboring nodes corresponds to the most recent common ancestor of those two leaves. And then we could use the spacing between those dots and make that proportional to the age of that internal node. And just that very simple definition gives you kind of a good approximation of the tree. Subtrees that are very close, of really closely related sequences, come up as dots that are very close together, and then major clades separate from one another. Okay? So how could we extend that idea for the local trees? Well, let's, instead of drawing a dot, let's just draw a line and draw that line for the period of time, the length of the sequence for which this tree exists. And then as the tree changes, just adjust the order and spacing of the lines. And so here's a cartoon where we can see that in this region, there's two major haplotype groups of 3 and 3, and then we can see that this sequence has now joined the other haplotype group. Okay? So if we just apply that visualization trick to real sequence, we can see that same trend. We can see that here there are two major haplotype groups, basically two big subtrees with lots of leaves in each one. The red lines here are just correspond to angled lines here that are jumping from one spot to another. They occur because of recombinations. Over here, there's many different types of haplotype groups. Where there's lots of recombinations, that's a recombination hotspot. You can see how it really shuffles the number of ancestral haplotypes from one region to the next. What's kind of cool about this visualization is that it's an instance of the tanglegram problem. We have a local tree per site, and to really make the visualization work, we want to make one local tree differ from the previous as little as possible. This shows up with like gene tree, species tree reconstruction, also with like comparing species trees, and there's many algorithms for figuring out how to lay out trees to incur the fewest number of crossovers. This is actually a more complicated version of the problem because we have many local trees, and I've seen people describe this as the serial tanglegram problem. I currently have not tried to implement anything too fancy in these visualizations. I use kind of the greedy algorithm for laying out the trees. But it would be interesting to do something more principally. See if you could implement tanglegram problem, the serial version, efficiently. Also, I've mostly so far visualized particular samples from a distribution, but these distributions are diffuse. It's not really fair to look at any one sample. You want to look at like a consensus ARG, just like you would look at consensus trees in phylogenetics. But I have, maybe someone here knows, but I haven't seen a really good definition of a consensus ARG, especially one that plays well in a visualization like this. If you're interested in these types of visualizations, they are implemented in our ARG Weaver software on GitHub. There's programs that will implement this layout algorithm, the heuristic version of it, and then it can serve these visualizations over a web server. And there's even software that allows you to kind of align this visualization with the UCSC genome browser, so you can see how the hotspot track corresponds to lots of crossovers in your ARG. And you can click on any site in the genome in order to look at its local tree. Yeah, and just more examples of like a site that is under selection will have long haplotypes, and those show up as a region where the shuffling does not occur very frequently. Okay, so in terms of future directions, there's still a lot more to do to make the inference of ARGs even more efficient and to make them even more applicable for problems in demography. It could be used for doing things like phasing and imputation and local ancestry. And then I've shown you some examples of like how ARGs can be used for selection, but there's probably more summary statistics and things that could be done to detect selection. So I want to thank Adam Siepel, my postdoc advisor, and other collaborators from Adam Siepel's lab who worked with me on this project and did a lot of the data analysis, and I really enjoyed working with all of them. So that's what I have. I'm happy to take questions.
Thank you very, very much. That was, I can definitely say I understand ARGs better now than I ever have in the past. And ARG Weaver seems really fascinating. So
Thanks.
if anybody has questions, please do type them into the little questions box. Yeah, here you can see on Matt's screen. You can see there's a little Q&A tab. If you click on that, you can go ahead and put in some questions. So
Q&A. All right.
So yeah, my question was, can you describe a little bit in a little bit more detail about how branch lengths work for ARGs? I mean, obviously there's a whole collection of branch lengths from the local trees, but do people try to think about the branch length of an ARG itself?
So like adding up all the branch lengths?
Well, not necessarily. I mean, like when somebody talks about an ARG,
Yeah.
like does it have any notion of branch length?
I think you can define one, and I was showing one summary statistic that we could recover. Basically, you could think of so for a tree, we could talk about its total branch length as adding up all of its branches, the length of all of its branches, right?
Yeah.
And so for an ARG, we could extend that definition and just say add up the branch length for every site, because every site is still a tree. And so I think the units of that branch length are now different because you're multiplying by sites.
Mm-hmm. Yeah.
And so what does that number mean? The bigger that number is, the more variation you should see in that region, because more time has passed.
Right.
Basically, time times base pairs. So that's the units.
I really liked your visualizations, and obviously it's like being able to infer ARGs. Like if we could do it perfectly, would be fantastic. But I also wonder if, sort of, as you guys were working on that, you came along with sort of ideas for other summary statistics that people could use to infer demography, or if you, I mean, whether you think that we should be proceeding forward with trying to define and infer ARGs, and then using that as a foundation for inferences, or if you think that other summary statistics are warranted.
Well, definitely summary statistics are useful, and in many cases they do the job, and there's no point in making your life harder than it has to be. inferring the entire ARG is really making it a big challenge. It's going to be a lot slower. You're going to be limited based on length of sequences and number of sequences. So if your question can be answered with a summary statistic, definitely go that route.
Cool.
Yeah, but I do think the ARG can help in situations where your question really involves the topology. Things of like IBD and chromosome painting, these are kind of like lower resolution forms of the ARG. You are it's kind of like cutting off the top of the ARG and seeing what little subtrees remain. And then each subtree you say this is a block of common ancestry. This is an IBD region, if that makes sense.
Yep.
Yeah. And so inferring IBD is kind of telling you that one slice of all the local trees. So it's an interesting, ARGs can be an interesting perspective in terms of thinking about what you're actually seeing, what things could fool you with such regions. I don't know.
Cool. So we've got a question from Yaniv. He says, great talk. Can we use the ARG to look at the change in local or global recombination rate over time, e.g. a turnover in hotspots for the local case, or a change in the genome-wide rate of recombination relative to mutation during the evolution of self-fertilization?
Yes, so we did look at the ability to recover recombination rate. We mostly focused as variation along the genome, but yeah, you can definitely pose it as going a variation along time. That's pretty hard. So what we've implemented so far implements a very neutral prior. So in the prior of the ARG, there is we assume neutrality, and we assume either a uniform recombination rate or a given recombination map. And for some of our analyses, we used existing maps to define our prior. You can look at recombination nodes in your ARG samples, and that would give you a posterior. So maybe your posterior would show you certain trends of changing recombination rate along the genome, and it definitely is the case. Like those visualizations I showed, that was a naive uniform prior, but the posterior you definitely saw clumping of recombinations. Maybe you can see something over time, but it all depends on probably the balance of mutation and recombination rate. The closer that gets to 1, the less recoverable these things are. Maybe better methods could be done where you don't have such a naive prior. Maybe you parameterize it with that allows variation in recombination, for example, and then you could estimate that more directly.
Just following up on that, Peter asks, and I think you've answered this, ARG Weaver does use a constant population size prior, right?
Yes. Oh, wait, no. Piecewise constant.
Ah, okay.
Each time interval is allowed to be different.
So you can, it can infer such a thing, like changes in ancestral population size, as long as we think of them as piecewise constant.
I think the model can do that. We did briefly look into that, doing something like, the PSMC model and various other ones that try to estimate changes in population over time. Is currently implemented. That is also a fixed prior. You can specify a certain change trajectory in population size, and then what we've done so far is just look at the posterior of coalescence rates, because coalescence rates correspond to population size. And even though you used a naive prior, perhaps the posterior you could see changes in population size. But that is probably not the most powerful approach. It would be good to parameterize the prior and then in your MCMC sampling, sample over those parameters, which is much closer to what other methods do, where their goal is to estimate those changing population sizes. That they explicitly parameterize that and sample over it or estimate posterior means of them, things like that.
Right on. I guess I'm curious, like, this is a completely different parameter regime, but we like to think about HIV and other viruses that, well, other things that recombine. I mean, would it be insane to try seeing what ARG Weaver does, or is the parameter regime just so different that?
I don't know off the top of my head how it would work. I think the parameter that matters the most is the ratio of recombination with mutation rate.
Which unfortunately is quite high for
yeah, so that could be a showstopper. Also, gene conversion violates assumptions. I don't know if that becomes important.
Yeah, it's cool. I'm just thinking, yeah.
So, like for HIV, maybe you're interested. Yeah, it depends on what you're interested in. There could be simpler versions of this method that focus just on topology change. If you're looking for breakpoints, if you're just trying to get major topologies out, instead of doing stochastic traceback out of this HMM, you could do Viterbi. And that could be a way of getting min args, which is another problem people are interested in. Find the arg that has the fewest number of recombinations. It's a parsimony version of the problem. You could use a form of ARG Weaver to solve the min arg problem.
Yeah, I guess that's the other thing I was going to ask is if you do just want an MAP solution, is it still best to just jump around using your MCMC and then pick the best one?
Yep. Yeah, and I did make a version of this to find a minimum recombination graph. There are ways of choosing what branch to remove that is most likely to remove recombinations. So, over the iterations of the MCMC or your proposals, you want to try and remove recombinations, get smaller and smaller. There are ways of picking branches that seem to be not placed very well. They're always jumping around too much. There are ways of identifying them, removing them, and resampling those. And that kind of targeted resampling is no longer MCMC. It violates all the rules you're supposed to follow, but it is a way of minimizing recombinations.
Cool. Looks like Peter has one more question, which is for both his and Yaniv's questions. You suggest computing observed minus expected numbers of coalescences and recombinations. He'd like to know if that's an easy thing to do.
So the observed is very easy to count. So when you sample the ARG, the output will be lots of ARGs, and each one is a sample from the posterior, just like in phylogenetics you have a sampling of trees. And so you can write scripts that walk through an ARG and count whatever you're interested in. So here would be count the number of recombination nodes, and you do that for each sample, so now you have a posterior distribution on that count, on that statistic. And that would be what you observe. And so then you could compare that to what you were expecting, if I'm understanding the question correctly.
I didn't understand it any differently, except maybe he was wondering if you could do it, yeah, more directly. Like if there's some way of incorporating the expected number in that would make it easy to compute this distance or something, or this difference. But okay. Well, like I say, that was a phenomenal talk. And let's see, the next one is going to be in about a month. It's going to be from Peter Ralph, who was just asking lots of questions, and about his tools for working with ARGs. So thank you very, very much, Matt. This was, like I say, an excellent seminar, and looking forward to the next one.
Yeah, thanks for inviting me, and I look forward to the next one too.
Great.